Like other nucleic acids, deoxyribonucleic acid, or DNA, is a chain of nucleotides. Each nucleotide contains a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogen-containing base. The order of the DNA bases determines an organism's traits by causing it to produce specific proteins. DNA contains 2 chains of sugars bound to phosphate, twisted into a helix. The bases stick off to the side and join the bases of the other chain by hydrogen bonds. The shape of the bases allows adenine to bind to the base thymine, while guanine and cytosine will always pair. This base pairing causes both strands to contain the information needed to determine the sequence of the other strand. The cell uses this to precisely copy the DNA for new cells, a process known as DNA replication. Replication starts as an enzyme called helicase unwinds the DNA helix and separates the 2 strands. Another enzyme, DNA polymerase, then comes in and adds new bases to each strand, following base pairing rules. The result is 2 new DNA molecules that are identical to each other and identical to the original molecule. Each contains 1 strand from the original molecule and 1 new copy. The new DNA molecules remain attached together at a region called a centromere and coil around proteins in the nucleus to form chromosomes. The DNA copies will separate into new nuclei in a process called mitosis. Humans have 23 pairs of chromosomes, with 1 set from our mother and 1 set of 23 from our father. The order of bases in our DNA can be used in identification because it is unique to each individual, with the exception of identical twins who start from the same fertilized egg. The most common method of DNA fingerprinting uses STRs, or short tandem repeats. These are regions of DNA that do not contain instructions for proteins. Instead, they repeat the same sequence of bases over and over. Each person can have 0, 1, 2, or many repeats of an STR at a particular place or locus on a chromosome. While 2 people might happen to have the same number of repeats at one locus by coincidence, scientists typically look at 13 loci. There is only about a 1 in a billion chance that all of these will be the same between 2 different people. DNA fingerprinting starts by copying the DNA so there is enough to analyze. The regions of DNA containing the STRs are copied using a process called polymerase chain reaction, or PCR. PCR gets its name because it uses the same enzyme, DNA polymerase, that the cell itself uses for replication. The enzyme was isolated from hot springs bacteria to allow it to stand up to repeated heating. A primer section of DNA is needed. Primers base pair near the STRs to get copying started in the right locations. Finally, PCR requires new adenine, guanine, cytosine, and thymine bases to add to the DNA. These components are placed in a machine for an automated sequence of steps. First, the DNA is heated to separate the strands. Next, the sample is cooled to allow primers to bind. Finally, DNA polymerase copies the DNA, resulting in 2 copies. This is repeated. Heat to separate, cool for primer binding, and copy to make 4 strands. Another repetition gives 8 strands. In a couple of hours, over 1 billion copies can be made. Once the STRs are copied, gel electrophoresis is used to separate them on the basis of size. The DNA is loaded into a gel and a charge is applied. Negatively charged DNA moves through the gel toward a positive charge. Longer STRs with more repeats tend to get tangled and move slower, while those with fewer repeats make it to the end of the gel. This gives a quick visual pattern showing the length of a person's STRs because fragments of the same length travel the same distance. The STRs can be used to match a suspect's DNA to the DNA at a crime scene, or can be used for paternity testing. Because we inherit half of our chromosomes from each parent, we also inherit half of our STRs from our mother and half from our father. We pass on one of these STRs from each locus to our own children. Children's DNA must be found in one of their parents. If an STR does not come from mom, it has to come from dad. This is how we determine paternity.